Welcome everybody. My name's Christy Spire. I'm the team leader of nature education here at Green Adelaide, and I'm very excited to be here with you this afternoon at our second instalment of our seminar series for 2024. I'd like to acknowledge this amazing country that we're probably all of us meeting on. There might be others coming from further afield. Ngadlu Tampanthi, Ngadlu Ghana, Yatanga, Imparenti. And it's good that we acknowledge that we're meeting on Ghana country today. And we feel very privileged to work with our local Ghana elders and the wider community of Ghana and Aboriginal people here in our midst, and to learn a lot about them and their country all together. It's a very privileged experience that we all have to be a part of. And we know that many of you are working with them all across Adelaide as well. So these seminar series are all about sharing the great work that's happening across Adelaide to help make it cooler, greener and wilder and more climate resilient. These sessions are being recorded and will be available on the Green Adelaide YouTube channel afterwards. We know there's a stack of people that might not have been able to make it this afternoon. You are invited to ask questions at any time by putting them into the chat. So we don't have a vocal function today, and we will answer as many of these as we can at the end of the session. So today we're launching into the power of citizen science. Citizen science has a long history in South Australia, spanning various fields like bats, birds, frogs, fungi, dolphins and dung. It provides vital data for scientific inquiries and insights unattainable by researchers alone. As technology has advanced, citizen science has embraced digital platforms, replacing traditional handwritten notes and logbooks with apps and digital databases, providing greater opportunities for participation. So beyond the contributions to science, citizen science also fosters nature connection and is a great way for community to learn about the world around them. Last year, Minister Cloast announced a $2 million Citizen Science Fund to support a new online platform and programs that will help improve South Australia's biodiversity knowledge. So in today's seminar, we're hearing from Jason van Weenen, the team leader of Urban Biodiversity for Green Adelaide, and Helen Owens, the Scientific Officer for Biological Data for the Department of Environment and Water. And they're going to delve into real-world examples of showcasing how citizen science can empower researchers by expanding data collection. They're going to unravel the mysteries of what happens to data generated on key databases and apps in SA. And they're going to demonstrate how to use these available databases and apps to identify flora and fauna. They will explain how data on rare and vulnerable species are protected and discuss any ongoing challenges and opportunities for us to all participate in. So my slides are not going through to show you the overview of today's session, but basically we've got 40 minutes with Jason and Helen, and then we've got about 15 minutes at the end for some interactive questions. So please pop them into the chat if anything comes up throughout the conversation, so that we can make sure that they're answered. Otherwise, we'll follow them up for you after as well. So I'm going to hand over to Jason and Helen now, and hopefully your slides will move a little bit faster than mine. So over to you.
Great. So
excellent. Take it away.
I guess I'll start. I've been fortunate to work in the biodiversity conservation space for quite a long time, and when I think back about citizen science, it's basically something which I feel I've sort of had an opportunity to play a part in right back before my uni days. I used to go out with departmental rangers at the time, and we'd go out and do a lot of hooded plover surveys and all these sorts of things, which when I think about it, it was all done on that ranger's spare time, and he was doing it voluntarily, and so getting that exposure to getting out there and collecting data and contributing to science about our biodiversity sort of when I think about it, it's sort of stems back from quite a long time ago, and I guess it extends through a lot of the work that we sort of do through the project work we do today. We're sort of connecting in with a whole variety of citizen science data and seeing that come in, but also getting involved ourselves wherever we can.
Yeah, and I've been with the department about 30 years, but throughout that whole time, certainly in my spare time stayed involved with citizen science, different organisations, and in my current work, heavily involved in trying to make sure all that information flows to the right people. I was thinking about when Jason and I first met, and it was like about 30 years ago at the South Australian Herpetology Group, back when we didn't call that sort of stuff citizen science. It wasn't even really cool to be part of a group like that. But things have come a long way now, and yeah, the stuff that Jason and I learned being part of that citizen science group back then has, like, you know, we've carried it through to our department work ever since, and it's helped us no end. So yeah, we've always been passionate about citizen science, even before it was a thing. All right, so when we started looking at what to present today, we thought we better define citizen scientists. We looked up a few different definitions that are on the web. Everyone has their thoughts on how what a citizen scientist is, but we were disappointed with some of them, like using words like amateur or non-experts, and we sort of, you know, discussed that. We thought, well, it doesn't really suit what citizen scientists are these days. We came up with this definition of, basically, it's volunteers helping the science across all levels of expertise, but basically means that you're, you know, contributing to science of your own free will and without expecting any pay. But yeah. Well, Christy just went through all the outline of what we're hoping to cover today, so I won't read all them out again. And we'll just move straight on to the first one.
Real world examples. Okay. So I guess there are a whole range of different ways that citizens contribute to really meaningful science programs, which follow through to a whole range of different delivery at the end of it. So here's one example, for example, where information on a whole variety of vertebrate pests. It's a database to capture those records. A community can contribute those. Pick a species like a deer, for example, record opportune deer sightings or fox sightings, and with these applications, these researchers and departments at the receiving that data can then assess the information coming in and plan programs around that. And we've seen that here in SA, where some of the information coming in, for example, on feral deer populations around the state have been, that information's been picked up on and helped with the delivery of on-ground programs to manage and protect really important areas from deer impacts. Just some other examples. So here in Green Adelaide, we've got a whole variety of projects, and we are regularly calling on the help of citizens out there to contribute information to help us deliver the projects. That square-tailed kites, for example, there were only there's not many nests in the state, fewer than 10 birds in South Australia, and so when we've been trying to find nests to look at the some of the things impacting or affecting this species, and so we called on the public via Facebook call out, and we were very fortunate to find another nest in the Green Adelaide region, which basically doubled the number of nests that we had to work with. So quite significant, very small numbers of birds, and so that was fantastic. Likewise, we've been using community calls to help us locate Escapee Alexandrine parakeets. They're an invasive species, and we're trying to find out where they are across the landscape and reassess or make sure we have a good assessment of the current risk that they pose to our native parrots and potential invasion risk. And so that's been a really helpful way to get those observations coming in. Things like black-chinned honeyeaters as well, very rare in the region. We often go through the bird observation sites, whether it be on Facebook, South Aussie Birding, those sorts of sites, to actually look at the records that are coming in and see where birds are in the landscape. We'll have a manual process of getting those observations and following them up and try and get them into databases. So it's a project-based activity where we actively go and get those records and try and get them entered into databases. But we also use information. Penny Payton does a chinwag newsletter, which actually tries to capture all those observations as well to help with their conservation. And the moth I've got on there is just another example. We've got projects which are being delivered around invertebrates, which stem from some of this citizen information that's coming through on a whole range of different species nowadays.
Yeah, a few other spunky citizen science apps that we were just going to showcase. This Echidna CSI, which is one of the ones that probably has had a lot of publicity and a lot of people have heard about. So it encourages people to send in sightings of echidnas through an app, and it also has this extra component of getting scats sent in. So I think you can actually pick up plastic bags and that's off them to have like what you need to send in echidna scats if you find them. So this has resulted in a high quality voucher data set that can be used for people like this example up in the top corner showing you a scientific paper that's come out of the work that's been done on the scats, looking at gut microbiome, I knew I'd get that wrong, biomes for wild and captive echidnas. So that's pretty cool that science papers are coming out, and you can see Echidna CSI has been acknowledged there. And this citizen science project was also used for like post-fire monitoring on KI, where the echidna subspecies is actually threatened. So that was pretty cool to be able to have that resource to tap into. So that's a really good one. Another exciting one is the Megan Murray-Darling Microbat project, which is run by the Murraylands and Riverland and Landscape Board. And it's been going, it was run for a few years and worked by people who were interested could register and pick up a bat detector and put that up in locations that they were visiting. So they got around, as you can see, all the locations there. They got around a lot more places than the researchers may have been able to. And in the 2 years that project was running, it collected more records than had been collected over the previous 100 years. So that was pretty cool. It just shows you the power of citizen science in what sort of quantity of information it can bring in. And also through the study, 2 new locations were found for the threatened large-footed myotis that just came from a couple who were travelling on a houseboat and decided to register and put up a bat detector. So that's really exciting stuff. Another amazing one that's been around for a while is actually comes out of Green Adelaide with Steve Walker's work on the Frogwatch SA project. And there's lots of great stuff been coming out of that over the years that it's been running. And last year, this Cosmos magazine article that Rupert Mathwin put together showcased like how important the citizen science component has been for frog data in South Australia. You can see that those bits I've highlighted. The article mentioned that citizen science data is essential for filling the gaps, and that 18,000 records are from areas with no routine monitoring programs normally. And that citizen science have increased the number of records from January to April by more than 1,000%, which is just mind-blowing. And according to Rupert, 87% of South Australians' frog calls were recorded by citizen science apps like iNaturalist, Frogwatch, and Frog ID. So that's pretty cool. Is this blocking the screen for people down the bottom, do you think, Jace? No.
Hopefully not.
No. Let's put it up there. All right. Okay, a few more examples. Yeah, there we go.
Okay, so yeah, this is just another example. A lot of the information that's coming in now is on a whole range of things like invertebrates and butterflies are included there. Interestingly, we've got lots of great invertebrate information in the South Australia Museum, but a lot of it isn't available, hasn't been mapped. The records are all there. But what we're seeing now with a lot of the apps and data coming in through a whole range of means is that the databases out there are now able to produce maps, and that's enabling us, because of all this citizen science, or a lot of the citizen science information, we're able to do status assessments of species, which we haven't done in the past, or haven't been able to do in the past, because understanding the distribution, the extent of populations and things are very important parts of assessing the status of species. And so with these maps, it's enabled us to do a lot more in that space. And so we were able to assess all the butterflies in South Australia over the last couple of years, and that's been really fantastic because it's highlighted some species which are of concern, and we can sort of support the nomination of those species to help look after them better. So yeah, a great example of just the amount of information coming in.
Yeah, so we could go on all day, couldn't we, Joseph, with like citizen science
example.
Yeah, examples have just been amazing, but that's just a few to get people inspired. So yeah, now we'll just go into the mystery of what happens to all the data that's generated by all the amazing citizen scientists out there. So yes, just showing all these coming in. It might be giving people the heebie-jeebies because one area of citizen science is like the confusion with all the apps that are out there. Is one of our major challenges. So that's just a small sample of some of the main apps that people will jump up in front of them. Yeah, and there's just a lot out there. So it's confusing for citizen scientists, but it's also confusing for us data managers in working out what data flows where. So a big part of a big tool that I use in my job is to sort of try and map the data flows of all these different apps and make sure the data is ending up where it should so people can access it. So I'm just going to introduce you to a little bit of data flow stuff. You know, in an ideal world, I'd love it if all of these apps on their front page had a little flow chart that showed the where the data was going once you hit that submit button. But some of them do have parts of that, but others it's quite a mystery and takes a bit of, you know, contacting people or researching to work out where the data is actually going. But we are getting better at all this. All right, so just to introduce you to a few bits about the flow of species data in South Australia. Now, we couldn't do this without introducing to the biological databases of South Australia, which is the area I've worked in for many years. So I'm a bit biased, but biological databases, which we call BDBSA, are hosted by the Department of Environment and have been around for about 30 or 40 years. And they endeavour to bring together all the research grade data for South Australia and get that out to the right people. So if you haven't heard of BDBSA before, we do have this dashboard that showcases what's like how much data is actually in the system currently. And you can see that there's like over 6 million records of flora and fauna from over 1,300 projects.
So just with that, Helen, a lot of that information has come in from surveys, but when you mention projects, a lot of other projects, like if there's a university study on a particular species through the research permit, that data flows back through as a project, and so that's helps to contain it all in the one area. Yeah, for sure. We encourage data to come in from any projects around the state, like NGOs like NCS and Field Nat groups who are collecting vetted data and submitting it, and also consultants doing work for mining companies and things like that, and Landscape Board and the department staff obviously contribute a lot of data in this space as well, and yeah, through anyone getting scientific permits and wildlife ethics. So we have quite a few sources, but it focuses on sort of vetted research grade data. So that's on our department website, that dashboard, if anyone wants to sort of drill down a bit further and learn more about what the biological databases hold. So just to show you how data flows, just to introduce you to the idea of data management and data flows. Basically, we've got some BDBSA templates and tools that let researchers upload data into BDBSA, and then the BDBSA data flows out into a departmental mapping tool that's available on the web, sort of free of charge to the public, that's called Nature Maps. So that's a really cool tool if you haven't been in to look at that. It's only the data that doesn't have any restrictions on it that can flow out into that public space. But we'll talk about sensitive data and restricted data a bit later.
So we send the data out to Nature Maps weekly, and we also send a feed out to the Atlas of Living Australia quarterly. Now, if you haven't heard of ALA or the Atlas of Living Australia, that's like a national version of our Nature Maps. So Nature Maps is SA focused, but ALA is national focused, and it tries to bring all sorts of data sets from citizen science and research grade stuff and institutions all together in one spot, so it can be discovered and accessed in one spot for Australia. So it's amazing. It's been around 20 years or so, and it's a great tool that we promote and lean on. And then a bit more obscure, but some of you might have heard of this GBIF, which is the Global Biodiversity Information Facility, and that's like ALA on steroids at a global scale. So that's just an international repository that tries to do what ALA does, for across the world. So that's amazing. Like, it's just, incredible what we can do these days compared to 20, 30 years ago. All right, so that's when we map it out like this, you can start thinking about these different environments as whether their main focus is about collecting data or their main focus is about managing the data, like BDBSA, it's a management environment, or if the main focus of these tools is delivery. So we tend to say Nature Maps, ALA and GBIF are delivery tools because they help us deliver the species data out to people that need to use it and want to access it. So when we start looking more at all these variety of citizen science apps that I put up before, you can start thinking of whether you can start getting your head around all the different names you hear and think, oh no, that is a collection tool, and the data from that collection app or whatever will flow into ALA or BDBSA or wherever it's going. So it helps you work out where your data's going and where you can access it. All right, so then if we introduce something like iNaturalist, which is a community-based app that allows the collection of records. So its main focus is about collecting data. It's an international app, isn't it, Jace?
Mm-hmm.
Jace is a bit addicted to it.
Yeah, I'm a bit addicted. Yeah.
But it's pretty cool. So its management is done by the community, basically. So it's and all the data from iNaturalist flows into the Atlas of Living Australia. And then you can sort of see here how the iNaturalist data currently does not flow into BDBSA. So if you were looking in Nature Maps for iNaturalist data, you won't see it. This just helps you sort of work out some confusion sometimes. But if you want to go to Atlas of Living Australia, you'll be able to look at the iNaturalist data and compare it to the BDBSA data that's getting fed in as well. So it depends what you're trying to achieve and what you want to do as to what tool might suit your needs.
And just in terms of some of this management, so I guess what happens with the BDBSA system is that there are a whole variety of processes which are related to the checking and verification of the data, making sure that all the important information with the records there before it's to make sure it can be used most effectively. And likewise with this community-based management example for INAT, it's where the validation and so it's a community process where the different observers and experts on INAT are helping with that validation and checking process. So it's just a different process happening in those two different areas.
Yeah. So if we take INAT away and we bring in some of the other apps like that you may have heard of, like eBird, FungiMap, Feral Scan, Echidna CSI, and BirdLife, then it's always of interest to sort of think, okay, once I hit that submit button, where is my data flowing? And I've got some question marks in the box there because often it's unknown or a mystery, or it might take a little bit to unravel where that's going. We try to get our head around each of them and make it flow where it needs to go. Things like FungiMap, Feral Scan and Echidna CSI are all now flowing into ALA. They weren't all for a while, but they're all flowing in there now, which is great. Mm-hmm. But though, it might be a 2-year delay or something, sometimes you're not sure. eBird, I think it is actually only available from inside the eBird app at this stage. I couldn't really find it on ALA last time I checked. And BirdLife, you can find it on ALA, but that's actually a subset of all the data BirdLife does hold, because they hold a lot of project data that they might not be public. So they do send all their sort of opportune records to ALA, and all the BirdLife data does actually flow to BDBSA, but we don't have the license to distribute it all through Nature Maps. So yeah, that's enough complications for you, but just to give you the idea that every sort of app's got its own, data flows that can be a bit tricky to unravel. All right. And then we can't talk about these without introducing the idea of the social media platform, which Chase has already touched on. But basically, places like Facebook that get records coming into them aren't really a proper collection tool, because they're not sort of set up with a structured way of making sure all the right bits of the information go with the record. So they're a bit random. Sometimes you've got people going in there pulling the good records out, but often the records that go in there just sort of, they bypass the collection and the management protocol, and they just end up in this black hole that's lost to science. So I don't want this slide to be too harsh, but it's really good for everyone listening today and all of us to try and encourage any records that are coming into those Facebook platforms to make their way to the appropriate app, which often might be INAT, might be a good spot for that. They have their place, these Facebook forums and that, to enthuse people and get communities talking and helping with IDs, but it's really good to redirect great records.
Yeah, one of the things you probably notice when you see a great record go into a place like Facebook is that it might have some of the data, and so with proper records there's usually a minimum data set, observer, the accurate location, the species, the date, those sort of minimum data requirements to make a really good record. And that's the problem when you look on Facebook, you see a great observation, it may be identified, but you might not have the observer, the date, the location, and so you can't really make the most out of that record. And that's a lot of the work that Helen does with cleaning data sets. And
I'm quite impressed, Joseph.
Yeah, I know.
Well, finally got through.
Yeah.
All right. So if you get it into the right spot.
Yeah, and this is the thing, and I guess one of the very cool things, having an appreciation of where the data, when you enter data, where it flows into the system and where it ends up, it depending on that location will determine how useful, how accessible it is, and to a whole range of different uses. And so here are some examples. Like we regularly use those data sets for status assessments, like the butterfly example, and trying to find populations, look at the issues that they might be facing and trying to figure out how to improve looking after them. There's a lot of planning, fire planning, land management planning, reserve planning, that the information is really useful to guide and make sure all the important considerations are being made for patches, and it happens all the different at so many different levels and basically all the time. So once these records are going in, if they are available and being shared to all, then it has all these flow-on benefits that many people probably aren't aware about. I guess one of the things is that when you think of some of the apps which might contain the data sets, but they might not flow through to these other broader data sharing platforms, is that the researchers will be able to still get their research projects out of them, but just some of these other benefits that you might hope to get might not be possible because of that limitation in sharing.
Yeah, but making it accessible is the main thing. Get it into one of the proper apps, and then what we call it is purposes unknown. You never know what it will be used for, but it can feed into all these great areas that are really good. So, yep. All right, next topic is how to use some of these apps, and you can use them for lots of different things, but today we were just going to briefly run through how they do like a species distribution map. A lot of them have lots of tools, but most of these are the sort of the main apps that we would use for looking for a species distribution with South Australian data. And all of these apps can do, all of these platforms or tools or whatever you like to call them, can all do species distribution maps. They can all do a species list for an area as well, if you want. And the ones you can see up here, are mainly delivery tools, apart from INAT, which its main focus is collection, but you can still actually do a species distribution map within it as well. They all have pros and cons. So nature maps, you can just go in there. It's just a free web app with no registration required, and it's got the curated research quality data for SA, and also has this added bonus of SA specific environmental data layers, which are really cool and detailed. Atlas of Living Australia obviously has national data, so that's a bigger focus if that's what you're interested in. And it has a lot of associated tools and information, including like images of the species you're looking at. INAT's obviously great for record collection, as we've mentioned, but if you're doing a species distribution map in there, it's only got the records that have gone into INAT, so you're missing out on anything that might be in nature maps. That might meet your needs for what you're trying to do at that point in time, so that's fine. It depends what you're trying to achieve. You also can't download the records out of INAT. So the other apps you can actually download like a table with all the records that you've selected. But INAT is global, so you've got all that global feedback coming in as well. And GBIF, which we won't talk much more about today, but it's like, as I said, ALA on steroids. So it's got the international scope if that's needed for whatever you're doing.
The other thing I just mentioned about INAT is that it's really useful for a whole range of species. It maps everything from a slime mold to an invertebrate to whatever. So that is a really handy thing I notice about that in terms of capturing, and provides an opportunity to map things which some other apps mightn't.
Yeah, so just firstly, nature maps. I won't bore you with the detailed training, but we do actually have another seminar that was recorded last week on nature maps training. All these apps you could do, an hour session or a whole day session on training people in them. So you can look out for that nature maps training one if you want more detail. But basically, we've set up these clever little flora and fauna tools up the top there that are your best option if you're just coming in to do a simple distribution map or species list for an area. They make it easy. You don't have to learn much about the app. We've also got this very groovy little 2-minute help video, which is really lets you know what you can do quickly, and then you can stop and pause and go through the steps a bit if you need some help. So basically, you just start typing the species you want, name or common name, and it'll narrow down a list. I've just picked Planigale today, which is this groovy little marsupial mouse that's found in the arid zone. I have to sneak in a few groovy pictures along the way. And then nature maps will give you a map of all the records, and then you can download them. Or you can zoom in and push on that record identity button to find out more about the specific record. So that's pretty simple little app. Does a lot more, but we're just covering that today. Also just quickly showing you the specific environmental layers that it has for South Australia. So this is cool. You can bring it up under your species dots and see, this particular species is associated with drainage lines and cracking clays. So you can start seeing that when you bring up the veg layer or a wetlands layer. So yeah, the layers in here are very well utilised and liked by consultants and people outside the department who gives them access. All right. Atlas of Living Australia. If you haven't looked in here, definitely go and have a poke, because it's very fun and it's on great, fast, groovy software that makes it, except on Sunday when it was not working, but no, it's good. Most of the time it's very, very fast and very good. So once again, you just type in your Planigale name, and it gives you all this associated information. Like you can see gallery there where you can go in and get images. You can arrow down the bottom there, shows you that it gives you a basic distribution map when you first come in, but you can push that view and download occurrence records, and that will take you into the detailed information about those records. So you'll get a more specific map. You can select the records tab and swap to the record list rather than the map view if you want. And
you can see you're getting the national view as well, so that rather
so it's not, once again, as we said, ALA's national, not South Australia. And it also has these groovy filter. It's got lots of good filtering of capabilities, particularly if you want to turn different different sources of data on and off. You might just want the museum records, or you might just want the iNaturalist records, and that you can filter very easily on this. But these filters up the top, they try to sort of idiot-proof it for us, and they drop out outliers and absence records and ones with poor location reliability. So they'll drop them out to try and give you a more accurate map that you presented with at first, but you can turn them back on if you want. All right, over to Jace for iNat.
Okay, so yeah, so as Helen mentioned, there are lots of similar functionality. Here's the Planigo again. Interestingly, not surprisingly, Planigales are discovered more during surveys where you're actually putting pitfall traps and research projects, and so they're not the easiest things to come across. And this example highlights how if you look on iNat, you pull up a distribution map, you might go, wow, I've just discovered the first one in South Australia. But what you do is you jump into ALA and actually look at the records there as well, and that would help put that record into perspective where you've got that broader collation of records in ALA. So that's just something to keep in mind. I find I jump between multiple apps when I'm looking at distribution or answering particular questions. So here's another approach where you can just go and explore areas like Helen mentioned. You can look at all these records. Again, these are just ALA records, so lots of them out there. You can put a polygon over a particular area and you get a list of, there's 22,000 observations in that zone, which I've created there, that polygon. And you can scroll through and look at all the species. There's 2,800 species there. It's interesting when you look at in some of these examples of the things that's been recorded. It's great because you're seeing all these different groups, cat sharks and squid, and there's some there listed as plants. So these are just some examples of some of the more recent records that have been added. But it's great to see those records added as plants. So someone's entered a record, they're not sure what it is, but they've gone broad because they don't want to put in the wrong species, and that's really important if you want your maps to be remain correct, because you could say it's a particular plant, it turned out to be the wrong one, and your distribution maps, which you will be incorrect until someone goes and corrects that. So it's great to see that broad classification, because it's not a problem if it's listed as plant. And then again, you can use the same area. You can actually sort out. So I've just gone and said, okay, what are the butterflies and moths in that zone? And then you it cuts the list down, so you can have a play like that, and narrow it down to species in an area or whatever. I guess, as Helen mentioned, iNat is also a data entry tool, and so because so you can look at maps and things, but you can also, when you're entering records, you can it's got some interesting functionality, like here's a moth, and you can compare it to the moths which have been seen in the area. So if you're not sure, you can actually have a look and see what else everyone else has observed nearby, which is useful when you're trying to figure out what you might have seen.
Very cool, isn't it? All right, just getting down now to how a lot of this data comes in, and people are often worried, I've seen something, but it's rare, or, sensitive.
Sorry,
Yeah,
I'm just going to go back. I just would like you're saying with the training on for nature maps and things like that. There is training for iNat, and I haven't done the training myself, so I feel like a bit of an imposter. So I wouldn't. There's so much more functionality. I'm sure there's people who know the app a lot more than me.
We only dabbled in the species distribution side of those. So we'll just have a look at the way we manage sensitive data that can't go out so much. For reasons of risk. So most of the apps we've been looking at deal with sensitive data by this sort of method, where they round down the coordinates, and when you put the species up that's sensitive on your map, you'll see it's got it like snaps to a grid. So that gives you an indication that it's probably been rounded down a bit because it's high risk data. People call this obfuscation or denaturing or
fuzzing.
Well, I was getting to that because that's the latest word for it, fuzzing, and that's my favourite. So yeah, they fuzz data, and we fuzz the coordinate, and then we also remove the location comments, so you can't sort of go to someone's area. So you'll see that as the standard way across Australia for fuzzing sensitive data most of the time. In South Australia, we try not to fuzz much. So we have this process where species that are considered sensitive can be nominated, and then we toss up whether the risk of fuzzing the data so that people won't be able to go and collect it or whatever is a greater risk than the risk of actually withholding the data, and then people who like are doing roadside veg spraying or developments in areas and mining clearance and stuff can't get access to the data. So we have to weigh up those two risks and decide what category the species falls into. So yeah, some states just do a blanket rule and apply it to all their threatened species, but in South Australia we sort of assess anything that's nominated and try to keep the list low as possible. We've got about 200 at the moment, and as you can see out there, most of those are plants because of the issues around orchids. But we've also got some things that like they're not sensitive for a conservation reason, but they're sensitive for like a toxic fungi that might be used to, you know, that's dangerous for humans to be getting hold of too much of that. Or it might be an associated species, so the plant itself's not threatened, but it might support butterfly colonies that are, so then we might fuzz the plant species data for that area or something as well. So yeah, on our website you can look into more about that and you can see the list that we do fuzz. But just rest assured that if you're putting your records in, we're keeping track of that, and any records you put in, if they're considered sensitive, will be fuzzed even if you give us the accurate coordinates. So yep. And INAT does it a bit differently.
Yeah, well, INAT, so I guess INAT is consistent in the way that if things are listed in South Australia as sensitive, well then that when things are identified, well then that denaturing should happen, the fuzzing should happen through that whole entire process. But there's also opportunities for people to put their own privacy, buffer the records themselves, so fuzz the records themselves just so the exact location isn't showing up, or you can completely hide the location if you want to not put the record on to public view that location until you feel confident that's going to be okay.
All right, we're just moving into winding up now, Jace. So we're going to discuss the challenges and opportunities that are sort of front of mind for Jason and I in the way that we deal with citizen science. So, well, taxonomy is one that drives me or keeps me busy, I should say. Mm-hmm. Yeah, it's a challenge for everybody, especially when we're applying that to records in databases. So updates on taxonomy that are being done, and also differences in taxonomy across jurisdictions and like different states and that calling things different names. So this causes confusions and is a challenge. It's an ongoing challenge. There'll be no fix. But yeah, all states on that are all working towards a national species list. So that's great news, and hopefully we'll get there one day.
And my thought, well, I just put some pictures up of some undescribed millipedes that I've come across in South Australia.
Had to get them in somewhere, didn't you?
Yeah, I had to get them in somewhere, like these amazing critters, and we just don't have any taxonomists who are looking at them, and so you get to a dead end sometimes with observations. So taxonomy, it can make it challenge when you're citizen science to enter things, because there is, you know, the IDs may have not been.
It's a tricky world.
Validation is obviously a very important thing, making sure you validate getting those validations, and it's a real challenge. There's amazing people out there doing a lot of work around validation, but there are limited number of bods to do it and lots of records coming in. Confusion.
Confusion over what app to use. Well, I covered that a bit earlier. It's basically I say to people when they ask where do I put it, I'm like, pick the one that suits your needs the best. And like, if you love fungi, go to fungi, you know, use the fungi apps. If you like birds, some of the bird apps are great. If you're a bit of a generalist, INAT's great because you can put all sorts of taxa in there. So there's no right app. It's more about picking an app that has good data management protocols and that you like, that you can use, that suits your needs. So try them, stick with the one you love, just make sure it's going in the right direction.
I guess the sustaining engagement from citizen science and app managers. I mean, one of the things that if you're not familiar with the apps, you might enter a record hoping for an ID and it might not happen, and that's for a whole variety of reasons that may be based on the quality of the photos, the information that's being provided. And so that feedback, the lack of feedback, can be potentially a challenge for some people out there. And likewise, we've got a whole range of different apps which might be part of a research uni project, and once that project wraps up, those apps may still be sitting out there, and there may not be as much management. So some of those, it's potential that you come across some of those apps out there. And sleep deprivation.
Yeah, you can talk about that.
Yeah, it's like they're just addictive, these apps.
Not everybody stays up all night putting records after records,
yeah.
But Jace does, so he's very sleep deprived.
It's a bit of a challenge to capture all these cool records. Yeah, that's right.
And not get addicted. Yeah. All right. So opportunities though. Yes, so advancement in apps is just amazing. Like every time I see Jace, he tells me about the latest advancement in an app and how cool it is. So it's really hard to keep up with, but it's really amazing. And the stuff that's going around, AI and that is just making like helping people become a lot more experts in areas and that, and just making data validation a lot easier for us. So that's really exciting area and opportunity. I mean, what's to come? And yeah, what have you also?
So even just with that, like an example with the AI stuff, with that photo there, amazing moth, but the app might even suggest the name of the plant that the moth's sitting on. So it's that clever that it's giving you a couple of suggestions around the moth and the plant. So it's just mind-bending, which is very cool. So it makes it a lot easier to add records and get suggestions and learn. You get to connect with a whole range of experts. So the apps provide some amazing opportunities to connect directly with some really knowledgeable people out there who are also the citizen science often volunteering their time to contribute, and they can be from anywhere in the world. So it's amazing the number of people all over the place who are providing information.
Yeah, I mean, previously they might have been, just stuck behind their microscope in a little, dodgy room in a institution somewhere, and now they're sort of connected with people all over the world and getting all that knowledge flowing out. And so it's great. Yeah. And then the other great opportunity with citizen science that is the endless enthusiasm that we've got to harness. So like there's no end to how many people are out there that are getting involved, and it makes collecting data, like, it gives us a whole new perspective on just what we can do. So that's great.
I'm just mindful of time, and we've got some, page up, page down. So yeah, just some of the take-home messages that spring to mind. It's always great to consider where your data ends up. So that's around that maximising the impact. So if you can think about where that information flows, like Helen showed some of those information flows, it's certainly encouraged that getting the information where it's most accessible or that it's publicly available is fantastic.
Yeah. And just being mindful that there's a limited number of people who are out there doing the validation of records. So, you might not get feedback in a week or two, but 6 months down the track, you might have found like a new species for the whole state or something like that, when someone does get the time to come to it. So it's just being patient and aware of how the whole thing works.
Yeah.
And as Jace will put there, it's a privilege to be corrected. Like
it's
not criticism. It's not great to have this feedback and community sort of discussion stuff. So that's great.
Yep. Have a play. It's one of those things where I think using the apps and seeing what they're capable of is a really good way to go. That's how I sort of feel I got started, and I know there's lots of other apps that I haven't touched as well. So just recommend having a play. And obviously, finally, this massive thank you to everyone out there who are, supporting and contributing to these apps, the collection of data, biological data, and helping with verifications and observations, because it's unbelievable. We really value the data.
We're just blown away by the citizen science help out there. Yep. We're almost just like embarrassed sometimes when we put up a project and overnight it's all sorted out for us by citizen scientists, and we're just like in the office and we're like, oh my goodness, they want another one today. We're going to put up another one. Yeah, so it's crazy. So thank you.
Okie doke. Thank you very much.
So good. You've done well to pack in so much information in a short period of time. Well done, team, and within so much enthusiasm, and hearing your personal stories and how you come to it as well is really cool. So I think there was quite a few questions before you showed that lovely flow chart, because people's minds were kind of getting blown with where the data goes or doesn't go. But I think there was one just confirmation on whether ALA data goes back into BDBSA. Does it go back and forth, or is it just because there was some confusion about that? But if we can just get that cleared up, otherwise we can send it through. I think you might have answered it, but yeah.
Oh no, not really, because there's
not directly.
Everybody's got their own specific question about that, and our focus in the department is to make sure data's accessible. So fantastic. The ALA data is accessible, whereas we concentrate on getting stuff into BDBSA that wouldn't be otherwise. So it would be lovely if all the BDBSA data was merged with ALA in one environment completely. I mean, it sort of is in ALA, but no, we don't bring everything from ALA back into BDBSA at the moment. We're concentrating on getting, the bird life stuff that's not, and the data sets that aren't making their way to ALA. And the research data from universities and that's not being uploaded into ALA, it's coming to us. So yeah.
It's a good one to remember that there are so many data sets and constantly discovering amazing data sets that haven't been placed into these corporate databases or sharing databases, and so there's a lot of work that you've been doing just gathering those and just even reporting that the types of data that are still yet to get added is huge.
Yeah, it's like a complex web, isn't it, of its
Yeah.
Awesome. There was one question about the best app for marine life, which I'm not sure was touched on.
Well, ALA is pretty INAT's being used. I see INAT's being used a lot for a whole range of marine taxa stuff. Yeah, so it's certainly a lot of information getting entered, and I know Janine Baker here in SA has promoted the use a fair bit of INAT, and through sort of been working on projects to get information in, and so lots of cool records and discoveries coming in through that platform already.
Yeah, I think I just saw Rosalie put up there that there's a lot of specific projects on INAT, and obviously Jason, I haven't got a head around all of them, but some specific marine ones by the sound, so that's great. So if you've got an area of interest, look for relevant projects on INAT. And you'll have a whole community there to join in with.
Awesome. Yeah, and Rosalie's also put up another tip for help on iNaturalist. Go to the Ferox Australis website, and that's the Australian iNaturalist community. Yeah, so there's interactions there. Is that right?
As we said, there's training resources for everything we've talked about today. So we were just trying to give a bit of an overview, but there's so much more we want to show everyone about.
So much more
inside all the apps.
Yeah. Awesome. Someone confirmed there's several marine projects on iNaturalist. Lots of great presentations, so everyone's really happy. I think all the flowchart questions were actually answered back at the top. I'm just going into one that I think I might have missed. Oh no, other people were providing lots of information. There's a whole community online already of answering each other's questions for you guys. So you're off the hook a little bit. Thanks. People from the Australian Citizen Science Association. Thanks, guys. Linking people to all of that information. We don't know it all by any means. We're just trying to get the data flowing. You did a great overview of it. Okay, thanks, Nat. There was one question. Does South Australia Government use ALA data and by extension INAT and other citizen science app data in equivalent ways to state biodiversity databases?
For sure. The thing is
that living example.
Yeah. So the thing is, if there's information on a species that's only in ALA or INAT, or INAT effectively means it's in ALA, but that we use whatever data you can, and that's the whole point about having it on available platforms. And you can so it definitely, sometimes it's the only location for the information. So you definitely use where, and I think that gets to the point that Helen was talking about, where as long as things are available, as long as your data is going into these available locations, that's the main thing. yeah, that I guess the focus with some of the work you're doing was trying to get more data, more of the stuff that isn't currently available into those same sharing platforms.
That's right. If someone says their data set's flying into Atlas Saloon Australia, we probably put the stops on it there and don't bother putting it into BWSA, and we'll get some other data that's not available anywhere else into BWSA. Certainly Green Adelaide obviously use it, and Threatened Species Unit, I know they don't do anything without looking at our data and then adding any extra stuff from ALA, which incorporates iNaturalist and Feral Scan and Echidna CSI and all the other stuff. So yeah, but it's got potential to be used wherever people look for it.
Yeah, excellent. I haven't found, someone said, I haven't found this to be the case for several states, so it's great to hear. Another question, should we start recording IBIS?
IBIS,
it's a trick question on it.
Well, hopefully there is.
I think that's a trick question.
Yeah, well, hopefully I'm sure there's lots of IBIS records getting in, and there's so many different. I'm just thinking there are big city bird apps. Yeah, that's right. That would be rad.
We didn't.
Thanks, Brad.
Yeah, we didn't talk about that though, Jace, that common species are good to record too, like not every time at every bin. But it's
I think that
like don't rule them out. Like when I was trained in survey methodology, it's like, the first day you write down every kangaroo, every sleepy lizard, because you never know when it's going to decline or what it's going to be useful for. So yeah, it's no harm in putting in the common stuff too.
Definitely important to do so, I reckon. Yeah.
Brilliant. And Jack was asking, is there a chance we get access to download INAT data if we keep plugging its use?
Well, I'm happy that it flows into ALA and we can grab it from there. So I feel happy that it's just getting into a system and shared through that way. So that's because it's very quick. So once it goes into INAT, I think it's fairly a short turnaround time before it's appearing in ALA. So so I can wait a few days.
We'll let INAT concentrate on their spunky AI tools and stuff, and
for sure
ALA can do the downloads for them. So it works.
Yeah, brilliant, awesome. Excellent. Well, I think we've covered off on everyone's questions. So thanks again for your time and putting that presentation together, Team Jason and Helen, and thanks to everyone that's joined us online today and those that will join us later as well. So from here, we will send you an email with some relevant links, and we'll include some information on, there's some upcoming iNaturalist training happening, and then there's also the International Citizen Science Initiative coming up, the City Nature Challenge, which runs from April 26 to 29. So if you're new to it all and want to jump into something soon, that's a good one to jump into. And our next session in our series is It's Not Easy Being Green with Dr. Stefan Redlek on Tuesday the 23rd of April as well. So we'll send you all a registration details to that with links, and also the recording from today as well. So we hope you can join us at the next one and get out there and get enthused by the power of citizen science. It's been an awesome session today. Everyone take care, and we're finished right on time. Thanks everyone.
Perfect. See you. Thanks everyone.
Thanks team.